Okay, so good afternoon, ladies and gentlemen. I would like to welcome you at our today's webinar that we are organizing together with Promega Germany, and it will focus on the NanoLuc-based technologies for studying protein biology. Before we start, I would like to just quickly introduce our company to those who are not that familiar with us. So eSport is an exclusive distributor made of Promega and Lonza on the Czech market, and we also supply other producers such as molecular devices, cytosmart microscopes, or some consumables from Alpha Laboratories. Part of our service is that we provide technical support for our assays or the assays that we supply to our customers, and we are also open to organizing smaller on-demand webinar just for your laboratory. For example, if you are interested in some specific topic that includes Promega technologies, feel free to contact me or any of our colleagues on our email or our company's email, eastport@eastport.cz. We are also active on Facebook and Twitter, so you can start following us there. And finally, I would like to quickly introduce our today's speaker, who will be Dr. Erik Bonke from Promega Germany. He's been working in Promega as an application specialist in the field of luminescent reporter assays. So he's the best person to ask any of your questions regarding reporter technologies. So I would like to give word to Erik now, and I hope you will enjoy the webinar.
All right. Thank you for your introduction. So let me briefly share my screen. Yeah, please let me know if you can see it.
Yes.
Okay, perfect. Okay, so welcome everyone. Thanks for joining this webinar. Thanks for your interest in this topic. Today we're going to focus on the so-called NanoLuc Binary Technology platform, or NanoBiT platform. And essentially this is a complementation reporter that we mainly make use of to study all kinds of things ranging from protein-protein interactions to quantitatively looking at dynamic processes that take place within cells. And today we're going to look at three of the applications that we utilize this platform for. I'd like to start by saying a few words about the company Promega. I guess the majority of you probably knows that we are renowned worldwide as the bioluminescent company. And this is because we have gathered a huge expertise in developing these luciferase-based technologies over the years. So meanwhile, we have a history of around 30 years in developing these technologies. And initially, this started by utilizing the firefly luciferase, which is probably the enzyme that most of you are familiar with. Later on, also the Ranilla luciferase, which we mainly utilize for gene reporter experiments. And then in 2012, there was basically a breaking point, because at that time Promega developed its own luciferase enzyme, which we call the NanoLuc luciferase. And on this particular enzyme, many of our current technologies are based on. As a brief introduction, what NanoLuc is, it is a recombinant luciferase that we actually derive from a much larger luciferase, multimeric luciferase complex that in nature you can find in this deep sea shrimp depicted here in the top left panel. And this luciferase is actually a tetrameric protein, so it has these 235 and 290 kilodalton subunits. It's a 106 kilodalton protein complex. And during the course of studying this enzyme, the distinct subunits were isolated, and this blue 19 kilodalton subunit was actually found to possess the catalytic activity. However, as indicated on the slide, once isolated, this subunit almost lost the enzymatic activity, so we only have a small signal output when we add the substrate of that luciferase. And this is basically the point where Promega came into play. So we took this 90 kilodalton subunit of the oplophorus luciferase and we subjected it through an evolutionary process by site-directed mutagenesis to end up with what we call the NanoLuc luciferase. And furthermore, we also developed or co-evolved the substrate that this enzyme converts. And we came up with a substrate that we called furimycin. So this this two-component system now based on what we call the NanoLuc luciferase and the substrate furimycin now is pretty efficient in a way that, as you can see, it allowed us to boost the light output of the system by a factor of 2.5 million. So pretty impressive. And in essence, this means that the signal generated by this enzyme really is exceptionally strong. So if you compare it with the conventionally used luciferases such as Renilla and Firefly, we have an about 150-fold higher specific activity with NanoLuc, which now allows you to really set up sensitive or highly sensitive assays with this enzyme. But also, if we look at the molecular weight of NanoLuc, it is beneficial because it's way smaller than these other two luciferases. So it's about half the size of Renilla and about 1/3 of the size of the Firefly luciferase. So with the 19 kilodalton, it is one of the smallest luciferases that are available. And this makes it in particular useful when you want to use it as a fusion reporter. So if you want to fuse this luciferase to a given protein of interest, then of course the smaller the better, because that helps you to avoid any artificial effects that a reporter might have on the fusion partner protein. So today we're going to focus now on 3 technologies that make use of the NanoLuc luciferase and that are all found on this NanoBiT technology platform. And I will show you in the next slide what this NanoBiT technology platform is. So we're going to look at the use of NanoBiT for studying protein-protein interactions in live cells. I would like to introduce you to one of Promega's newest technologies, the so-called Lumit immunoassays. And in the second part of the talk, we're going to focus on the hybrid protein tagging system, which is a pretty cool bioluminescent peptide tag that you can append to your protein of interest and therefore render it detectable and enables you to run quantitative analyses with this protein of interest. So what's NanoBiT? NanoBiT is an abbreviation for NanoLuc Binary Technology. So in other words, it's a 2-component system based on the NanoLuc luciferase, or more easily expressed, it's a split variant of the NanoLuc luciferase. NanoLuc was split asymmetrically into a relatively large subunit, 156 amino acids, and then a tiny peptide subunit, 13 amino acids. The unique thing now is, which is really unique to all commercially available split luciferases, is that we now further optimized the properties of these 2 subunits. The large subunit was optimized for its stability within mammalian cells. What we ended up with is a subunit that we called large bit. And for the peptide subunit, we actually ran a whole library screen to identify peptides that differ in their affinity for this optimized large bit subunit. peptide that we identified, we subsequently called small-bit, because this 11 amino acid subunit now shows an extremely low intrinsic affinity, meaning we have a very high KD value, 190 micromolar, which means these two subunits, when you have them in solution, they will not spontaneously complement. They will only complement when brought and held into physical contact by other means. So we thought that this configuration, large bit and small bit, is ideally suited to perform analyses of protein-protein interactions in cells. And the idea of this NanoBiT PPI, or protein-protein interaction system, which will be the first technology that I'd like to present today, is that you fuse the two proteins of interest that you would like to measure the interaction of. So in this image, protein Y and protein X, you fuse these two proteins to either one of the split subunits, large bit and small bit. And the fact that we have relatively small tag sizes now minimizes any artificial effect that a reporter or a tag might have on the fusion partner protein. If the two proteins now interact, we achieve a physical complementation of the two subunits, which reconstitutes the functional luciferase, and we can measure this by detecting the luminescent signal. So now, due to the optimized affinity of that small bit subunit for the large bit counterpart, we can use this technology to really measure both ways. So we can measure the association of such an interaction, or we can measure the dissociation. And this is really something only possible because we have this optimized intrinsic affinity. You will not be able to measure in a reversible manner using, for example, split autofluorescent proteins such as split GFP. And with most available or commercially available split luciferases, you will also not be able to measure both ways, or if you can measure both ways, you somehow will have an artificial component to your signal, because the second major advantage of this optimized affinity is in fact that we massively boost the specificity of this assay, because a given signal that you detect in the assay is really due to the interaction of the two proteins that you would like to study, and it's not driven by the affinity of the two split subunits. And last but not least, the reconstituted luciferase, which we call the NanoBiT luciferase, is an extremely bright enzyme, which means you can work at low expression levels, because this enzyme allows you for high sensitivity, so you can really work at low expression levels, which in particular, if you want to study protein-protein interactions, and let's say you would like to study this interaction in a small subcellular compartment, then expression level really becomes critical, because at a given expression level, somehow everything will interact. And by keeping, or by the ability to keep the expression level low, you can circumvent these artifacts. Depicted here is the general workflow of this assay. It's a simple transfection-based experiment, and probably the most laborious step is the first step, where you have to do some cloning. You will insert the open reading frames of your two proteins of interest into the respective expression plasmid that allow for fusion to large bit and small bit. And what makes this step a little bit laborious is that we really recommend you to test all different combinations, meaning your protein X with large bit N and C terminally, together with your protein Y with small bit N and C terminally, and then vice versa. So depending on your proteins of interest, this will lead to 8 different possible combinations. There are some rare cases where you do not have to test all different combinations. For example, if one of your termini is processed, or if one of your termini is outside of the cell, or if you study homodimerization. So all these scenarios will lead to less than 8 different combinations, but in general you have to test these 8 different combinations and you go for the one that yields the highest signal to background ratio, and that's your combination that you utilize in an assay. However, once you've figured out the perfect combination, it's pretty easy. So you simply transfect the 2 plasmids. You allow for overnight expression of the recombinant fusion proteins, and the next day you proceed to add the substrate for the NanoLuc luciferase. You can optionally run a treatment of your cells to modulate the interaction, meaning to either induce it, or if it is a constitutive interaction, to block the interaction, and you simply read out the luminescent signal in order to conclude on the interaction of the 2 proteins. The second most important step when establishing such an assay is to control for specificity. I mentioned already that the specificity is improved by the fact that we optimize the affinity of the 2 subunits. However, you have to run a control to really prove that a signal that you may detect in the assay is really due to the specific interaction of the 2 proteins. And in general, there are 2 options that you have. First of all, you can use, as we call it, a tool compound, meaning a compound of which you know that it modulates the interaction. Or if this is not available, you can utilize negative control, which is included in the starter kits that we do provide. And this control is actually nothing else but an unrelated protein fused to the small bit subunit, and you express that fusion protein together with your large bit fusion. And provided that the signal of your particular interaction pair is about 10 to 1,000-fold higher than the signal of this negative control, then you can conclude on a specific interaction. It's important to note that this is not a rule. It's just a general guideline that is based on the experience we have made in the past by developing all kinds of PPI assays with NanoBiT. Another option that you have to prove specificity is illustrated on this slide here. So what you can do on top of these controls that I mentioned is that you simply co-express one of your 2 proteins of interest as an untagged variant, and if you titrate that untagged variant, then you should see a concentration-dependent decrease in the signal, as we can see it here, the blue graph, while this titration of the untagged protein DNA does not have any effect on this negative control. And the reason why we see this drop in the luminescent signal is basically due to competitive displacement. So if we increase the amount of the untagged protein, then of course the number of unproductive pairings, meaning your large bit fusion with, well, let's say the large bit protein X fusion with the protein Y without the small bit tag, because this interaction, of course, does not lead to an assay signal. So the more unlabeled untagged protein is present, the lower the luminescent signal should get in case it is a specific interaction that you detect. The model system that we initially validated the NanoBiT PPI technology with is the protein kinase A model, which is illustrated here. Probably you're aware of the fact that protein kinase A, or pKa, is a tetrameric protein composed of these 2 catalytic subunits here in green and these 2 regulatory subunits in purple. And in this assembled state, this enzyme is rendered to be inactive. However, if the cyclic AMP levels in cells rise, then cyclic AMP can bind to this regulatory subunit, which will now in turn dissociate and liberate the catalytic subunit, which is then rendered to be active. Cyclic AMP levels within cells are directly influenced by the activity of so-called adenylate cyclases, AC, which we can experimentally activate by treatment with forskolin. However, these enzymes are also indirectly regulated by the activity of GS-coupled GPCRs, such as the members of the beta-adrenergic receptor family. And these beta-adrenergic receptors can be activated by an agonist such as isoproterenol, or inactivated or blocked by an antagonist such as propranolol. So keep these three modulators, isoproterenol, propranolol, and forskolin in mind, because this is what we will be using in the next experiment that I would like to show you. So what we did here is that we expressed the catalytic subunit as a fusion to the small bit subunit and the regulatory subunit of pKa as a fusion to the large bit subunit. So in this inactive assembled state of pKa, we will have a bioluminescent signal due to complementation of the two NanoBiT subunits. However, upon increase of cyclic AMP and thus activation of pKa, we will lose this bioluminescent signal as the two subunits will dissociate. And this is essentially what we did in this experiment here. So here we transfected cells with the to express the two recombinant pKa subunits as large bit and small bit fusions. And now at different time points, we added these modulators that I just had on the previous slide. So the first modulator we added after 10 minutes is isoproterenol. So this agonist of the beta-adrenergic receptor indirectly increases cyclic AMP levels, and what we see is an immediate drop in the bioluminescent signal due to activation of pKa. This signal now recovers. That is what we would expect because of the activity of endogenous dephosphoresterases that break down cyclic AMP levels. And we can further enhance this recovery by addition of an excess of an antagonist of the beta-adrenergic receptor, the propranolol. So this simply displaces the agonist and therefore further promotes the recovery of the inactive pKa level. The important thing now is if we add forskolin at around 40 minutes, we now again see a massive drop in the luminescent signal due to reactivation of pKa. To prove that the signal changes we see here are really due to a change in cyclic AMP levels, we ran in a parallel approach an experiment where we transfected cells with one of our cyclic AMP glow sensors. So these glow sensors allow you to monitor cyclic AMP levels in live cells in real time. And as you can see, we have a perfect inverse correlation of these two signals. So whenever cyclic AMP levels go up, the red graph here, we see a drop in the NanoBiT signal, and when the cyclic AMP levels go down again, we see an increase in the NanoBiT signal. So this very nicely proves that in the NanoBiT PPI system, although we are actually working with tagged proteins, recombinant proteins, we are in fact able to modulate or to monitor, let's say, to monitor the biology as it is occurring within the cell without any artificial influence. So we are able to detect the activation and deactivation process of pKa. And furthermore, this experiment nicely illustrates that really the NanoBiT PPI system is working reversibly, so we can measure association as well as dissociation, which, as I mentioned already, is only possible due to the optimized affinity of large bit and small bit. What we actually offer here, besides the starter kits that are available through our catalog that contain the empty vectors, controls, and detection reagent. We also do offer a number of pre-established PPI pairs. As I mentioned, we generated quite a lot. These are just some of them depicted on this slide. So whenever you recognize that there is a distinctive pair that you would be interested in studying, then you can simply purchase this PPI assay directly. So you would not have to do the cloning, you would not have to optimize the orientation of the tags, so you will basically receive the two plasmids of the optimal configuration. For some of these pairs, we also do provide stable cell lines. In 99% of the cases, these are HEC293 cells. And finally, there's a so-called BBIT system available, which is in particular for those customers that know from scratch that they would like to generate a stable cell line. Stable cell line generation is not possible with the vectors included in the starter kits. So if you know now already you would like to generate a stable cell line, then you should go for this BBIT system. This system basically works the same as the conventional NanoBiT PPI system, with the exception that once you've identified your perfect combination, you can now fuse the two separate vectors into a single vector with a bidirectional promoter. And this vector now has a selection cassette, so you can generate your stable cell line by random integration, and you can select for appropriate clones using this selection marker. All these products that I mentioned are not available from our catalog. These are no catalog products, which means you will not find them on the website. So whenever you're interested in this, just give us a shout and we can provide you with additional information. Okay. So I illustrated the development of the large bit and the small bit configuration. We spoke about the use of this configuration for studying NanoBiT, studying protein-protein interactions using the NanoBiT PPI system. However, these two proteins, X and Y, could in principle be antibodies as well. And in this scenario, we will achieve a complementation of the two NanoBiT subunits by the two antibodies binding to one and the same analyte. And this is the fundamental principle that the Lumit immunoassays are based on, which is the second technology that I would like to introduce you to today. So the Lumit immunoassays, as I mentioned, are probably one of Promega's newest technologies. They serve as a powerful alternative to all conventional immunoassay approaches, meaning the traditional ELISA or Western blotting. I guess you're familiar with those methods anyway. However, you will agree probably that these are quite heterogeneous multi-step processes, because you always have to do several washing steps, you have to do a lot of incubation steps. So it's a pretty laborious strategy. And depicted here in the image is the workflow of a traditional sandwich ELISA, which, if you would start from scratch, starts by coating your microtiter plate with a primary capture antibody. Usually that's an overnight process. You wash away unbound antibody the next day. You block away free spaces on the surface. You wash, you add your analyte, your sample. You wash, you add your primary detection antibody. You wash again. You add your secondary detection antibody, which is usually coupled to an enzyme. You wash, and finally you add the substrate for that particular enzyme to read out the signal. So all in all, depending on what protocol you pursue, this is a laborious procedure that can take something between 1 or 2 days. By developing the Lumit immunoassays based on the NanoLuc luciferase, we were now able to significantly streamline that process. Because the Lumit immunoassays work in solution, we could get rid of the coating and the blocking steps. Furthermore, we removed all of the washing steps. And finally, we combined the 2 separate antibody addition steps into a single addition step of an antibody mixture. So as you can see, by these measures, we very much streamlined that procedure to end up with an assay that really can be accomplished very fast with an assay time of only 30 to 120 minutes. So these assays are not only fast, but their bioluminescent nature now brings all the benefits of bioluminescence to these assays. So they are very sensitive because you have minimal background, and usually we have a very broad dynamic range which allows you to mitigate sample dilutions in these assays. The fact that they are highly sensitive and that we have this homogeneous workflow, meaning we only have addition steps, now renders them also to be applicable to different plate formats, meaning 96 or 384 well plate formats, and it makes them also very easy to automate and applicable to high throughput applications. The Lumit immunoassays are available in different formats. First of all, there's a direct immunoassay format in which we make use of the primary antibodies that are directly labeled with the large bit and the small bit subunit. This strategy that requires labeling of the primary antibodies, we mainly validated for detection of everything that gets secreted from cells, such as cytokines or different peptide hormones. And as you can see, there is a long list of already existing ready-to-use assays for various cytokines and peptide hormones such as insulin and glucagon. So if these targets are among the targets you would be interested in, then this is a good way to be immediately ready to run an experiment. So these are ready-to-use solutions that contain all the required components, also the standard used for your calibration curve, etc. However, if you're interested in a different target or a different cytokine, then this is not a problem because we also do provide a labeling kit, which allows you to very easily, as you will see in a minute, label your primary antibodies of interest and then develop such an assay yourself. Secondly, there's an indirect immunoassay format. This is, as we say, more or less a generic format because here we make use of pre-labeled secondary antibodies. It's generic because you can, of course, use these secondary antibodies with different primary antibodies. So you can detect more than one target with these secondary antibodies. And of course, the benefit is that this approach does not involve any labeling. So you do not have to label your primary antibodies, which sometimes are rather precious. So you do not want to lose antibodies during the labeling process. Importantly, this format we mainly validated so far for studying phosphorylation events, so looking at intracellular post-translational modifications, so whenever we are working with cell lysates. And last but not least, there is a competitive assay format. So far, there is actually only one assay in our catalog that has been developed according to this format. This is the Lumit FcRn binding immunoassay, which I will not touch on today. But if you're interested in studying the interaction of therapeutic antibodies with FcRn receptors, then this assay is a nice choice. And according to the same principle, in the future, the near future, there will be numerous assays available that allow you to study the interaction of an antibody or the recognition of an antibody by various human Fc gamma receptors. In these assays, where you want to determine the interaction of an analyte with a target, in this scheme here, your analyte will interact with an antibody. Then what you need is to label the target with either large bit and small bit, and then to develop a so-called tracer molecule, because this tracer together with your target will form this bioluminescent complex, and you can now test for the binding of your analyte of interest by competitive displacement of the tracer, which will result in a decrease in the luminescent signal. Let's start with the direct immunoassay format, and here I really would like to walk you through the distinct steps that you would pursue when developing such an assay from scratch. The first step, of course, is the choice of the antibodies, and here Lumit is really flexible, so you have these three choices. You can develop such an assay by using 2 monoclonal antibodies, which is clearly the approach that will yield the highest specificity and also sensitivity. But on the other hand, you can also develop such an assay from only 1 polyclonal antibodies, which is clearly the most inexpensive variant. And in some cases, it will be the only option you have if there is a limited availability of antibodies for your target. And as always, there's a solution in between, where you have 1 monoclonal antibody and 1 polyclonal antibody. And this is in fact the configuration that you will typically find for ELISA, which means if you have a provider that provides antibodies for ELISA, this provider usually does provide so-called matched antibody pairs. So pairs of antibodies that have been tested in an ELISA, so they are working in an ELISA setting. And this is a nice option to make use of, so you can simply develop such an assay by using these matched antibody pairs. So once you've decided what strategy to follow, the first step is now to label these antibodies with the large bit and the small bit subunit. Also, similar to the NanoBiT PPI technology, also here we do recommend to test the different combinations. So your antibody 1 with large bit together with your antibody 2 with small bit, and then the vice versa configuration, meaning you have to test 2 different configurations, with the exception of the strategy where you only use 1 polyclonal antibody. Here, of course, there is only 1 configuration to test. The labeling can be easily achieved in a 2-day protocol using the Lumit immunoassay labeling kit that we do provide, which achieves really a highly efficient attachment of the large bit and the small bit subunit to the antibody, so an efficiency greater than 90%. And the principle of that technology or labeling kit is that you start by labeling primary amines on your antibody with a so-called amine reactive halotag ligand. So this ligand is composed of an amine reactive group, which is this succinimidine ester group here, SE, and linked to this group is a chloroalkane linker sequence, which is the sequence that is recognized by the so-called halotag protein in an irreversible fashion. So this sequence is irreversibly bound by the halotag protein. So after 1.5 hours of labeling, you will now remove the unreacted halotag ligands in a short buffer exchange step, and you now split your sample into 2 reactions. To one reaction, you will add purified halotag large bit fusion proteins. To the other reaction, you will add purified halotag small bit proteins. You now incubate overnight, and what is going to happen is that the halotag protein will irreversibly bind this chloroalkane sequence, and by this you irreversibly attach the large bit or the small bit subunit to your antibody of interest. The next day, you simply perform a verification of the labeling by running an SDS page followed by Kumassi staining, where you would like to observe this banding pattern at higher molecular weight. This is how it looks in reality. What you can see here is a labeling approach of a human interferon gamma monoclonal antibody. In the lane on the left-hand side, we did not any halo tag large bit or small bit fusions to the labeling reaction, so we only see this prominent band which corresponds to the unlabeled antibody. And then if we add halo tag small bit or halo tag large bit, we can see this banding pattern, this ladder at higher molecular weight, which corresponds to the X-fold labeled monoclonal antibody. As you can see, we also recognize these two bands at lower molecular weight, which corresponds to the unreacted halo tag large bit or halo tag small bit fusion proteins. But the good news is that you do not have to remove these from your labeling reaction because they actually do not compromise the performance of the antibody in an assay. To illustrate this, I'd like to show you this experiment where we detected a concentration series of purified interferon gamma using labeled interferon gamma antibodies. And for the red graph, we subjected these antibodies to a cleanup step, so we removed these unreacted fusion proteins, and we did not for the blue graph where we did not insert that cleanup step. As you can see, the performance is pretty much identical. So this nicely proves that the unreacted fusion proteins do not interfere with the performance of the antibodies in an assay. So once you've labeled your antibodies, the next step is that you work out the perfect combination, which is not needed for the approach where we have only one polyclonal antibody, as we discussed. And then for all three strategies, however, you have to work out the particular concentration of each of these antibodies. This is done in a matrix experiment as depicted here for a configuration where we use one monoclonal and one polyclonal antibody. So what you can see here is the monoclonal antibody fused to small bit and the polyclonal antibody fused to large bit. And we tested 4 different concentrations for each of these antibodies in the presence of the antigen. The same for the other configuration, the polyclonal with small bit and the monoclonal antibody with large bit. Again, the 4 different concentrations in the presence of the antigen. You then run the same reaction in the absence of the antigen because that helps you to determine the assay background, which is pretty low in the Lumit immunoassay. And this is due to the fact that we have, first of all, this optimized affinity of large bit and small bit. So we won't have a spontaneous assembly of the large bit and the small bit subunits. But on the other hand, the fact that in the Lumit assay we do always require 2 of the antibodies to bind the target protein in order to generate a signal, we avoid so-called cross-reactivity related signals that you will observe in a Western blot or an ELISA experiment. So if in an ELISA or a Western blot one of your antibodies binds somewhere else unspecifically, this will translate into a signal in the assay. In a Western blot, of course, you can see this because you can look at the correct band by comparing the molecular weight. But in an ELISA, this of course is not possible. In Lumit, always 2 antibodies are required to generate a signal, and the scenario that 2 of the antibodies bind unspecifically to the same target or the same protein is pretty much unlikely. So these assays are really specific. So once you've determined these 2 signals, you can now calculate your signal to background ratio, and you simply go for the configuration with the respective concentrations that yields the highest signal to background ratio. So in this case, we use the monoclonal antibody as a large bit fusion at 0.125 microgram per mil, and the polyclonal small bit fusion at 1 microgram per mil. With this configuration, you now run a first functional test by simply performing a calibration curve. So you will run your standard dilution in the plate, you add the mixture of the two labeled primary antibodies, you incubate for 1 hour, you add the detection reagent that contains the luciferase substrate, and you measure the luminescent signal. What you will end up with hopefully looks like this. So you have a nice calibration curve. In this case, this is an example for human interferon gamma. And this nicely illustrates the general attributes of the Lumit immunoassays. So we have a wide dynamic range, more than 3 log units, which makes these assays really sensitive. And also we have quite low lower limits of detection. So also with regards to the lower detection limit, these assays are sensitive. A nice side benefit of these assays is that you are flexible with regards to your assay design. So you can, let's say you would like to detect a cytokine, you could take aliquots from your supernatant at different time points and transfer them to a separate plate and run the assay there. Or you can directly add the assay reagents to your cells because the cells do not interfere with the assay performance. And I'd like to show you this by looking at this example here where we wanted to measure secretion of cytokines from activated T cells. So in this example, we were looking at so-called B-specific T cell engagers. These therapeutic molecules are actually fusions of 2 single-chain variable fragments of 2 different monoclonal antibodies, so that recognize different epitopes or targets. And the BiTE that we used in the experiment is called Blincyto. So this BiTE recognizes CD19 on the cancer cells, in this case the Rachi B cells that we used, and it recognizes CD3 on the effector cells that we added the CD8 positive T cells. Upon activation of these T cells, they secrete cytokines such as interferon gamma or TNF-alpha, and also they release granzymes and perforins that will now mediate target cell killing, so a specific killing of this cancer cell. So we wanted to know if we add our assay to this co-culture of these 2 cells, if we can measure the secretion of these cytokines. And as you can see in this example here, we are able to monitor this concentration dependent increase in cytokine secretion or in interferon gamma secretion in this case. So the more Blincyto, the more B-specific T cell engager is added, the more cytokines are going to be secreted, and we can nicely detect these in the presence of these 2 cell types. So the cells do not interfere. This direct assay format can not only be used to perform quantitative analysis of a target or an analyte, but we can use this format also for other purposes. And one purpose is to study protein-protein interactions in a biochemical assay format. One assay that we developed according to this strategy is the so-called Lumit SARS-CoV-2 spike RBD human ACE2 immunoassay, which is an assay that you can use to detect, on the one hand, inhibitory molecules, so molecules that block the interaction of the virus receptor binding domain with the host cell's entry receptor, the ACE2 protein, or you can also check for the presence of neutralizing antibodies. The principle of this assay is that we tagged the human ACE2 protein with a mouse FC and the RBD with a rabbit FC. If we now add antibodies labeled with large bit and small bit that are directed against mouse and rabbit IgGs, then provided that the two proteins interact, we can generate a luminescent signal in the assay. However, if we add any compound or antibody that blocks the interaction of the two proteins, this becomes evident by detecting a decrease in the luminescent signal. So this assay presents a nice alternative to the standard assay used to detect neutralizing antibodies and so on, which is the plaque reduction neutralization test. So this assay does not require any higher biosafety levels because we are not working with infectious viruses. It is fast to achieve, which is a benefit when compared to other surrogate virus neutralization tests that are ELISA-based. So we have this time benefit that we can accomplish this assay within hours instead of days. It is screening compatible. As you can see, it also has this homogeneous format, which you also will nicely see on the next slide. What I would like to mention is that in the near future, we will also be providing antibodies that are pre-labeled with large bit and small bit to detect various protein tags, meaning his tag, GST tag, or flag tag. So this allows you to kind of build your own biochemical protein-protein interaction assay. So what you need then is are your two proteins of interest fused to either let's say a his tag, protein 1 with the his tag and protein 2 with the GST tag, and then you can use our pre-labeled antibodies to establish such a protein-protein interaction assay and screen for example, inhibitors of this interaction. The workflow is pretty straightforward. It starts off by addition of your test molecule that you expect to inhibit the interaction. You now add your rabbit FC RBD fusion protein to this compound. You incubate for 30 minutes. That's an optional step, which is optional because with this you can maximize binding of your inhibitor to the RBD protein. You then add the mouse FC ACE2 fusion protein followed by addition of the antibody mixture. You incubate 60 minutes, add the detection reagent and measure the luminescent signal. So pretty straightforward, takes you roughly about 2 hours, so way faster than all ELISA-based strategies. Some representative data that was generated with this assay is shown here. So this is a titration series of recombinant ACE2, which is a is a putative strategy for treatment of COVID-19 because the purified ACE2 can now saturate the spike protein on the virus surface, so it can no longer bind or attach to the host cell. And what you can see here is that with increasing concentrations of this recombinant ACE2, we see a nice decrease in the luminescent signal detected with this particular assay. So it really blocks the interaction. The same can be done with different antibodies. So these are 4 different antibodies directed against the spike protein. As you can see, we can nicely work out the efficiencies of these different antibodies to block the interaction of RBD and ACE2. And what I did not have here on the slide, but the assay is capable of being used for this purpose, is that you can also analyze patient serum. meaning you can check for the presence of either neutralizing antibodies upon vaccination or post-infection with the SARS-CoV-2 virus. With this, I'd like to move over to the indirect immunoassay format before we're going to now then begin or move on to the short break that we mentioned at the beginning. So this indirect format, this generic format, as I mentioned, we validated for studying post-translational modifications such as phosphorylation. And due to the fact that we, with this format, mainly detect intracellular proteins, we named this format also the Lumit immunoassay cellular systems. So in any such experiments, you would probably start by treatment of your cells in case of phosphorylation to treat cells in order to activate a certain kinase that now will phosphorylate your protein of interest. The first step of the assay now is that you add a lysis solution which is digitonin based, and it is included in the kit, and you incubate for 20 minutes to release the protein content from the cell. You then proceed to add the mixture of the primary antibodies and the pre-labeled secondary antibodies that we do provide. You incubate for 60 to 90 minutes, add the detection reagent, and read a signal. So pretty straightforward, homogeneous workflow. To provide you with some flexibility with regard to the secondary or the reactivity of your primary antibodies, the species reactivity of the primary antibodies, we have the secondary antibodies, different secondary antibodies available that are pre-labeled with large bit and small bit. So we have different species reactivity. So you can actually use primary antibodies from rabbit, mouse, and goat. And if you have a different primary antibody, that's not a problem. You can also use the labeling kit to label your secondary antibody of interest. So in this particular example here, where we would like to detect our protein of interest and we would like to detect the phosphorylated form, we use one primary antibody that binds the phosphoprotein of interest, in this case from rabbit, and we use another primary antibody from mouse that detects the protein of interest, however, irrespective of the phosphorylation status. So with this particular antibody combination, we will exclusively detect the phosphoprotein only, because only the phosphorylated protein will be bound by the two primary antibodies, while the non-phosphorylated protein is only bound by one primary antibody. And as we discussed, this does not lead to the generation of the signal in the assay. How can you now normalize your data? In principle, there are two options. First, you can normalize on the total amount of your protein of interest. For this, you would need a third primary antibody, which you use to replace this phosphospecific antibody. So you replace this antibody from rabbit by an antibody that also binds your protein of interest, however, irrespective of phosphorylation. And you use this antibody together with this mouse antibody. This allows you to detect the protein of interest no matter whether it's phosphorylated or not, and you can now normalize on that value. The second option you have, which is also included in the kit, is the so-called cell titer Fluor assay, which allows you to normalize on the viable number of cells. This assay is based on a compound called GFAFC, which is a coumarine derivative that once within the cell is readily proteolytically processed by this so-called live cell protease into a fluorescent product. In dead cells, however, this protease is immediately inactivated, so no more fluorescent product is being formed. So what you can do now is that you add this cell titer Fluor reagent right away with the treatment of your cells. And in the moment that you add your lysis solution to the cells, you basically kill the cells. So you stop the formation of this fluorescent product. However, the product already being formed is stable up to 3 hours. So this gives you plenty of time to finalize the Lumit immunoassay first and then continue to read the fluorescent signal to normalize. I also would like to show you one representative data set which is focusing on the NF-κB pathway model system that we used here. For those of you not familiar with that pathway, it can be activated by various stimuli such as TNF-alpha or LPS. In all cases, this merges in an inactivation of the I-κB kinase complex, which will now in turn phosphorylate the inhibitory protein of the NF-κB transcription factor, the I-κB alpha protein. Once phosphorylated, I-κB alpha now dissociates and gets proteasomally degraded, while the now liberated NF-κB transcription factor can go to the nucleus and switch on transcription of NF-κB regulated genes. One of these downstream genes is the I-κB alpha gene itself, so that this de novo synthesis of the I-κB alpha protein now serves as a negative feedback mechanism to suppress NF-κB activity. In this experiment, we used 2 sets of primary antibodies to detect the total amount of I-κB alpha protein in green and then the phosphorylated form in red. We treated cells at time point 0 with TNF-alpha, and you can nicely see the time-dependent increase in the phosphorylated form, which is accompanied by a decrease in the total I-κB alpha protein level, which makes sense as the phosphorylated protein is being degraded. To check for specificity, we co-treated with a proteasomal inhibitor, MG132, and as we can see, this leads to an almost complete stabilization of total I-κB alpha protein levels, while we see an accumulation of the phosphorylated form. If we look at a greater time window, so this is a 30-minute time scale. If we look at 120 minutes, we can also visualize this de novo synthesis of I-κB alpha. As you can see, this starts by around 30 minutes, and then we see a stepwise recovery of the protein level, and we can block this by co-treatment with cyclohexamide, which is an inhibitor of protein biosynthesis. The Lumit immunoassay cellular systems we meanwhile validated for more than 20 phospho and total protein targets in various cell types, so this makes us very confident that this is a universal assay that you can actually apply to all kinds of post-translational modifications, or if you just would like to detect the total protein, that's also a possible application. The bottleneck, of course, is that you require the appropriate primary antibodies for your target of interest. Listed here are the targets that we already validated, and for all of these targets we provide application notes that are available for free download from our website, and these application notes do contain the respective protocol that was established and also information on the source of the primary antibodies. So in principle, if your target is among these listed here, you basically have a ready-to-use assay with the restriction that you have to purchase the primary antibody somewhere else. And with this, I would like to move on with the break. I think we mentioned 5 minutes. Well, if you have any questions in between, feel free to ask them now, or we can do that at the end of the presentation. Okay, so I hope everyone had time to grab a cup of coffee. We will now continue with the presentation, moving on to the third and last technology based on NanoBiT that I would like to present today, which is the high-bit protein tagging system. This brings me actually back to a slide that we already had before, where I spoke about the development of NanoBiT and where we ran this peptide library screen to identify peptides that differ in their affinity for the large bit counterpart. Another peptide that we identified in the screen we subsequently called high bit because this peptide had an extremely high affinity for the large bit counterpart. So if we look at the KD values, instead of the 190 micromolar we had for small bit, high bit now has a KD of 700 picomolar. So a very high affinity, meaning if you have these two subunits in solution, they will spontaneously complement and reconstitute a functional luciferase. So we therefore thought high bit is ideally suited to be used as a detection tag, and this is exactly the idea of the high bit protein tagging system. The idea is that you append high bit to your protein of interest. You have a very small tag, 1.3 kilodaltons in size, so this again minimizes artifacts of a tag onto the fusion partner protein. The nice side benefit of this small size, however, is also the fact that we can very easily and efficiently knock in the high bit encoding sequence using CRISPR into the genome, which now gives us the opportunity to really study in proteins at their native expression levels, while also maintaining the transcriptional regulation of these genes, basically, because these genes are still controlled by the native promoter. And in the end, that's a nice tool because it helps us to avoid so-called gene dosage effects that we sometimes do observe when ectopically expressing proteins using non-regulated constitutive promoters. The fact that we have a small tag is actually nothing exclusive to high bit. You will agree there are many other fusion tags out there, MAC, MUC, FLAG, HA, whatever, that are also around the 1 kilodalton size range. The unique thing about high bit, however, is that all these other tags do require antibodies for detection, and high bit does not. So high bit can be really within minutes be detected using one of the detection reagents that we do provide that you simply have to add to your sample. So again, it's a homogeneous workflow. And all these detection reagents do contain the purified large bit subunit along with the luciferase substrate and the buffer. So this detection protocol now really allows you to save a lot of time because you can avoid all these laborious steps associated with antibody-based detection, as we discussed earlier. And the homogeneous workflow now makes these assays amenable to high throughput applications and makes them also easy to automate. We do offer different detection reagents. First, the NanoGo hybrid lytic detection system. Here we utilize a lytic buffer. So if you add that to your cells, you essentially lyse the cells and detect the total amount of your hybrid tagged protein. With the extracellular detection system that uses a non-lytic buffer, you restrict detection to the extracellular space, meaning you can detect hybrid tagged proteins secreted into the cell culture supernatant or membrane proteins, provided that hybrid has been put somewhere in the active domain of these proteins. Why does this work? It works because the purified large bit subunit cannot penetrate the cellular membrane. So you essentially can only detect extracellular hybrid with this detection reagent. Then there's the blotting system that you can use to detect hybrid tagged proteins on a conventional Western blotting membrane, again bringing the benefit that you do not have to block, you do not have to excessively wash and incubate, you simply add the reagent to the Western blotting membrane. And finally, there's a strategy that supports live cell analyses. Remember, large bit cannot go into the cell, so in this case, you would have to have the cell express the large bit subunit themselves. So in this strategy, you will co-express the large bit subunit either transiently or stably along with your high bit tag protein, and then use one of our different nanoglow live cell substrates. Depending on the time frame you're interested in, you can choose from different solutions we offer. Probably the most important factor when speaking of a detection tag is that this tag provides a high specificity and selectivity and sensitivity, and that it's quantitative. And we think all these criteria are met by HiBit, so we have linearity across a broad dynamic range, more than 7 log units, and it's also quite sensitive, reaching down to the attomolar range, which in particular when studying proteins at the native expression level is something crucial. How can you now tag your protein of interest with HiBit? There are in principle 2 strategies. First, you can go for ectopic expression using an HiBit entry vector. We do offer HiBit entry vectors that allow for N or C terminal placement of HiBit to your protein of interest. There's also a variant that adds an N-terminal IL-6 secretion sequence so that your HiBit tag protein will be subsequently released from the cell into the cell culture supernatant. And we have these vectors available with different constitutive promoters to achieve different expression levels. However, HiBit is simply small, so 33 nucleotides long, so you can very easily and effectively rely on the expression plasmids that you may already have generated and add HiBit using PCR amplification. So that's also an option you have. The second strategy is to do this endogenous tagging approach using CRISPR. And here we developed the protocol that is completely cloning free, so no molecular biology is required because you can simply have these components needed synthesized, or you can readily purchase them. The protocol is based on 3 distinct components. First of all, you need a guide RNA, which is composed of your target-specific component, the CRISPR RNA that you have to design, and then a so-called trans-activating CRISPR RNA, which hybridizes with the CRISPR RNA, which is a universal component. So this is one of the reasons why we're going for this 2-part system instead of a single guide RNA, because you can order this tracer RNA in larger amounts and use it with any CRISPR RNA. The second component is a purified Cas9 endonuclease, because what we do is we assemble this RMP or ribonucleoprotein complex in vitro, and together with the third component, which is a single-stranded donor DNA, which is composed of these 2 homology arms that flank the HiBit sequence, we transfer these components into cells. Preferentially, you do this via electroporation. That simply yields the highest efficiency. And then subsequently, you can actually relatively early start to validate your tagging events, because you can detect HiBit with such a high sensitivity. So you can start 24 to 48 hours post-transfer of the RMP and the donor DNA. What we offer here is a do-it-yourself protocol that is freely available from our website, and we can also provide a free service. So if you are not experienced with CRISPR at all and you would like to tag a gene with the HiBit encoding sequence, then you can get in touch with us and we can make a proposal of 3 different CRISPR RNAs and also the donor DNA, so we can aid in the design of the target-specific CRISPR components and make this proposal. We cannot give any warranty that you will be successful with these. However, it may be a good starting point for those of you that are not familiar with that technology yet. Furthermore, we offer a long list of ready-to-use cell lines in which particular genes on particular gene backgrounds have already been tagged with HiBit, but also other reporters. And we offer these as edited cell pools and edited cell clones. How does the tagging workflow look like? It's pretty easy. The first step, as I mentioned, is the design of the CRISPR-specific components that are distinct to your target. And in this example shown here, we would like to add the hybrid encoding sequence right to the C-terminus of the protein encoded by this gene here. So what we need to do is to add the hybrid sequence right before the stop codon. To do this, we screen the region around the stop codon plus minus 50 nucleotides for the presence of a so-called PAM site or protospacer adjacent motif, which typically for the Streptococcus pyogenes Cas9 is NGG. And you will want that PAM site to be as close as possible to your desired integration site, so in this case the stop codon, because the closer, the higher the knock-in efficiency will be. Once you've found the PAM site, the CRISPR RNA simply corresponds to the 20 nucleotides upstream of that PAM site. The additional nucleotides needed to hybridize with a tracer RNA will be added by the vendor that you buy the CRISPR RNA from. And this is now needed to allow for Cas9 recruitment, which will now insert a double-strand break 3 nucleotides upstream of the PAM site. So 2 things to consider. If possible, you should design a CRISPR RNA that spans your desired insertion site so that you prevent recutting one once the hybrid sequence has been inserted. And also, you should choose a PAM site that allows for the double-strand break to be inserted outside of the coding region whenever possible. Next, you need the donor DNA, which simply corresponds to the 50 to 80 nucleotides upstream of your desired insertion site, the 50 to 80 nucleotides downstream of your desired insertion site. You should, when possible, mutate the PAM site within the donor sequence to prevent cutting of the donor itself or recutting of edited alleles. And then you insert the hybrid sequence in between. Once you've done this, you can validate your tagging events using the various detection reagents. This is shown here for the NanoGlow hybrid lytic detection system, where you see different signal intensities for different guide RNAs, and you would of course go for the guide RNA that yields the highest knock-in efficiency. Desirable for an assay is a signal to background ratio of 10, so that's a general guideline. If you have a cell line that already expresses large bit, you can also go for the NanoGlow live cell assay system, or you can go for the blotting system if you would like to receive information on the molecular weight. So after the tagging, what you receive is an edited cell pool, which, as I mentioned, is also something that we do offer. The edited cell pool, however, does contain a mixture of all kinds of genetic varieties. So you will have cells that will carry no mutation at all, heterozygous mutations, so where one allele is edited with hybrid, B-allelic mutations where one allele is edited with hybrid and the other may carry an indel mutation, and then also homozygous mutations. The nice thing is that only those cells that have integrated hybrid will show a signal in the assay. The others will not be detected. So what's the benefit of working with a pool which can actually be used directly for an experiment? The advantage of that pool is that putative indel mutation, for example, that has a negative effect on these cells is simply averaged out because you still have this genetic variability. We also see that the pools behave pretty much similar than an isolated clone in an experiment. However, of course, the bottleneck is that a pool is way dimmer than an actual isolated clone. So in this example shown here, a pool with a 5% knock-in efficiency would be approximately 5% as bright as an isolated clone. So it's not always possible to directly use the pool for the experiment. If you want to pursue, however, the isolation of the clone, the workflow is illustrated here, following your validated pool. So when you assure that you have the luminescent signal coming from your edited cell pool, what you do is that you expand this pool and then run a single cell isolation, either manually using limited dilution, or you can use a cell sorter. You expand these single cells to clones. You perform a copy of the plate, a triplet copy of the plate. One copy you use for the cultivation. The other you use to run the lytic hybrid assay to check which of the clones are luminescent. And for these luminescent clones, you use the third plate to isolate genomic DNA and do the genomic characterization. And all these results will now guide your selection of which clones to expand. I'd like to briefly illustrate what the hybrid tag can now be used for. These are just some of the examples, but I'm sure there are many, many more. First of all, you can use it to simply quantify or check for protein expression on the post-translational level, but you can also look at dynamic processes that your protein will now be implemented in. Customers have successfully used hybrid to study viral infection, and we just recently developed an assay to monitor the infection by SARS-CoV-2 in real time. You can look at protein secretion or the unregulated release of proteins from cells as it occurs during cytotoxic events. And finally, you can look at receptor biology, meaning you can monitor dynamic processes such as internalization and recycling of receptors, and utilize the hybrid fusion proteins to study the interaction of extracellular proteins or compounds with these membrane proteins. I'd like to show you one example which is focusing on protein stabilization. It is focusing on the HIF-1 alpha pathway, which I guess many of you are familiar with. It's a prominent transcription factor that is regulated by the presence of oxygen. So whenever oxygen is present, HIF is prolyl hydroxylated at these two positions, leading to its polyubiquitinylation and proteasomal degradation. Under hypoxic conditions, this oxygen-dependent reaction is inhibited, so HIF accumulates and now can go to the nucleus and switch on HIF-regulated gene expression. We can mimic hypoxic conditions chemically using phenanthroline, for example, and by appending now hybrid on HIF-1 alpha, we can monitor this dynamic process. What you can see in this example here is an experiment where we ran a titration with phenanthroline, and we basically used 5 different cell populations. In black, you have those cells in which we used the CRISPR approach to tag the endogenous HIF-1 alpha gene. And then we have 4 different populations in which we ectopically expressed HIF-1 alpha hybrid using a constitutive promoter, and we achieve different expression levels by using, first of all, different promoters and also transfecting different amounts of DNA. In all cases, we actually see a concentration-dependent increase in the luminescent signal. This nicely correlates with an increase in HIF-1 alpha hybrid protein level, as shown by this hybrid blot here. And we also observe an inverse correlation of the expression level and the fold response that we can detect with our assay. And in fact, we achieved the highest fold response in the cells in which the endogenous HIF-1 alpha gene was tagged with the hybrid encoding sequence. So when we express HIF-1 alpha hybrid under the native HIF-1 alpha promoter. And the reason for these are the so-called gene dosage effects that I mentioned at the beginning, because all these ectopic expression approaches will lead to an overexpression of HIF-1 alpha, so that this in the end leads to an overload of the cellular proteasome. meaning HIF1 alpha hybrid can no longer be degraded, and this now raises the background in the assay and therefore diminishes the assay window. By co-expressing the large bit subunit along with the hybrid fusion protein, the HIF1 alpha hybrid fusion protein, and using one of our live cell substrates depicted here, as I mentioned, we have different solutions for different time points of interest. We can turn this into a real-time assay, so you can monitor in real-time changes in the abundance of your protein with the nanoglow and uracil system up to 72 hours if you would want to. So this real-time measurements gives you additional information, and you don't lose any data because you really can cover the whole time frame. And in this particular case, the same experiment run with also additional downstream target genes of the HIF1 alpha protein, BNIP3, ANCA37, HIL-PDA. These are known downstream targets, which we also tagged with the hybrid encoding sequence. We can nicely see the timely pattern here. So we see upon phenanthrelin treatment at time point 0, we first observe an increase in HIF1 alpha protein level, and then this is followed by an increase of the downstream target protein levels, really emphasizing that these are in fact downstream targets of the HIF pathway. So with this, I'd like to move over to another application example where we looked at viral infection. I mentioned that we developed this assay to monitor SARS-CoV-2 infection in real time, the SARS-CoV-2 hybrid pseudotype virus-like particle assay. So in this assay, which is still not in the catalog, though it's available, we make use of so-called pseudotype virus-like particles. These are virus-like particles generated by using the HIV GAG polyprotein that we tagged with the hybrid sequence, and we pseudotype these virus-like particles by co-expression of the SARS-CoV-2 spike protein. So if we add these virus-like particles to our target cells that express ACE2, TMPRSS2, and also the large bit subunit, what is now going to happen? The particle will attach to the cell. This will lead to the entry of the virus, and now this leads to the release of the GAG hybrid polyprotein into the cytoplasm, which can now complement with the large bit subunit to yield a luminescent complex. So we can really measure the infection in the moment that it occurs using this assay. If we now add any inhibitor that blocks or interferes with the entry and fusion process, we can see this by a decrease in the luminescent signal. Here are two representative data sets. On the left-hand side, you can see a time course experiment over 5 hours where we added the SARS-CoV-2 pseudotype virus-like particles to the target cells, and you can see a nice time-dependent increase in the luminescent signal. And on the right-hand side, you can see the effect of neutralizing antibody directed against the spike protein, where we see a dose and time-dependent inhibition of the infection process by SARS-CoV-2. The last example that I would like to show you that hybrid can be used for is receptor biology. As I mentioned, we can look at internalization and recycling of receptors. In this example, we would express the receptor as a hybrid fusion, whereas hybrid is put at somewhere within the active domain of the receptor, and then we use the extracellular detection system that has the non-lytic buffer component. So with this system, you will restrict detection to the membrane-bound fraction of the receptor, while everything internalized will not be detected. In this example, we tagged the beta-adrenergic receptor 2 isoform with HiBiT, and we then treat it with the agonist isoproteinol. Again, we use different populations, cells that ectopically express the HiBiT fusion receptor, and then cells in which the endogenous gene was tagged with HiBiT in orange. And you can see isoproteinol concentration-dependent decline in the luminescent signal due to internalization of the receptor. Again, same story. The highest signal to background ratio is, or highest fold response, I have to say, is achieved with the cells that express the HiBiT fusion using the native promoter, in fact. And the same holds true for another example where we did this with EGFR tagged with HiBiT, and we treated with the EGF agonist of the EGF receptor. And with this, I'd like to close and thank you for your attention. You will be receiving the slides afterwards as a PDF file, so in case any question that we will not address now because of time limitations or whatsoever, or if it comes up later, that question, feel free to write me an email. I put my email here. And yeah, with this, I'm at the end of this presentation, and I'm happy to answer your questions if you have any.
Okay, Eric, I would like to thank you for a great presentation. So if you have any questions, you can either use the raise hand function or type it in the chat or unmute yourself, and you're welcome to ask anything. Okay, we have one raise hand.
Hello, can you hear me?
Yeah, we
Yes.
can hear you. Great.
Thank you for a very nice and clear presentation. I would like to ask, could you please say a few words about the mechanism of the reaction? Or if you know where is the binding site, if this binding site is at the interface of the subunits, or something more about the mechanism in general. Thank you.
You mean the NanoLuc luciferase reaction, or?
Yes, the conversion of furimazine. Or where is the binding site for furimazine?
If you're asking this with the intention, if there might be a problem that this binding site could get blocked if you fuse it to a protein of interest. If that's the intention of your question, we haven't seen anything similar. So I cannot tell you exactly the amino acid residues where the binding site is placed. I can send you a reference if you would like to the paper where this that describes the development of NanoLuc. But no matter where you fuse that enzyme to, you will have accessibility of the active site for furamycin. So that's not a problem.
Mm-hmm. Okay, but do you think that there is just a single binding site for this molecule?
Yeah, it's a monomer. NanoLuc is a monomer, and there's one active site, to my knowledge.
Mm-hmm. Because I'm thinking that I can imagine that the binding site is somewhere at the interface, because if there is fluorescence upon binding, so it's interesting. But as you said, there are some more information about this mechanism.
Yeah, are you talking about the protein-protein interaction assay, or?
In general, because I was looking for some information about the mechanism of this reaction, and I couldn't find some more details.
Yeah, I can enter the paper if you would like to read that paper into the chat. So you can download the paper that describes the development of NanoLuc. So So maybe you can find the information you're looking for in that paper.
Okay. Okay, thank you.
So 1 second. Yeah. Okay. Any other questions?
Like we said before, if you have any further questions that come up later after the webinar, you can send email either directly to Eric or to us in eSport, and we will answer your questions as soon as possible. Okay, so I guess if there are no other questions, I would like to thank you, Eric, once again, and see you later on some different webinar on another topic concerning Promega.
Yeah, you're welcome.
Okay, so thank you for attending and have a nice rest of the day. Goodbye.